on Mendel's genes, Pea Genetics. So please take it away.
Thank you very much. And thank you very much to the organizers for this wonderful meeting on this occasion. And hello, everybody who's online. So let me start my timer so I know where I'm going. Mendel's genes. What do we know so far? So by Mendel's genes, I don't mean Mendel's genes, I mean the genes of the peas. So I'm going to start with some introductory comments. Then I'll go on and tell you a little bit about the genes that have been characterized so far at the sequence level. And then I'll talk a bit about the uncharacterized genes and the current work that's ongoing in that area. So Mendel did use he studied many species of plants, 19 or 20 or more. And he published the 2 papers on plant biology. So here on the right is the 1870 paper where he made crosses in hawkweed and studied the hybrids that when they were self-fertilized and you look at the progeny, they stay the same as the hybrid. And in the following generations, they have the same phenotype as the hybrid. It's a constant phenotype. And so Mendel actually referred to those as constant hybrids. And that's because they have a different reproductive system to other plants. It's quite a rare reproductive system called apomixis. So those were his studies on hawkweed. And of course, I'm going to talk about his 1866 paper, where he studied peas and also beans and made crosses, generated the hybrid, which this time is called a variable hybrid, and self-fertilized the hybrid, study the progeny, and the progeny segregate for the traits he's studying. So they are called variable hybrids because the progeny are variable in appearance. So that's the paper I'm going to concentrate on. And you can read more about this in this paper here. Yes, pea was an ideal experimental plant for Mendel and others before him because there were so many varieties available, many different forms. They're very self-fertile, they're vigorous, and also because of the form of the flower. The flower envelops the reproductive organs and prevents insects from getting in, so you don't have any accidental outcrossing, which would muddy your results. So excellent experimental organism. And you can read more about the varieties that were available to Mendel in this paper at the time when he was working. Many varieties available on the market as commercial varieties. Okay, when you're crossing peas, what do you do? Let's not forget that peas are hermaphrodite. So it has male and female reproductive organs in each flower. And so when you're crossing peas, you will take, as the pollen donor, you will take an open flower that actually at that point will have already self-fertilized. And as the pollen recipient, which is going to be the female, you'll take a flower that's still a little bit in bud, like on the far left over there. And so you need to go ahead with the fertilization. So you need to emasculate the female flower. So you will take off the anthers, which have yellow tips there. And then you go ahead and pollinate with the pollen donor. And as you can see in this picture here, pea is really perfect for doing this. There's no need for a paintbrush. Pea, the female organ, is with the pollen on it, is a paintbrush because it's got hairs, and the hairs can carry all the pollen, and you simply, the female recipient with the paintbrush containing a presto, so that's the way to do it, and it's extraordinarily easy. Of course, you wouldn't do as much damage as I'm showing here. I just opened up these flowers to show you what's going on. And as we know, one pollen grain is sufficient to fertilize an egg cell, just one, and produce a seed. And Mendel was actually doing experiments to find this out. And you can read more about those in this paper here. Okay, so the fertilization has occurred and you've generated the F1 hybrid shown here. And it's more clearly shown on the far left there, the keel petals, which are fused and they completely contain those male and female reproductive organs. And so this F1 hybrid will self-fertilize, seed will be produced, they'll grown up to be the F2 progeny, which will segregate for the trait that you're studying, which in this case is the A gene, which conditions the difference between white flowered and pigmented flowered types. Right, so what were the traits investigated by Mendel and which ones do we know more about, a lot more about? So I'm going to talk about these 7 traits. And the first one we know more about is the R gene, which the wrinkled peas versus round peas. So peas, when they're dried down, they can be quite spherical in shape or they can have a wrinkled surface. And that's because if they have a wrinkled surface, they may carry a mutation in starch branching enzyme 1. And in this case, it's a mutation because of a transposon insertion into the gene. And that was found out back in the 1990s in that paper. So the R gene is encoding the starch branching enzyme shown here. When it's mutated, that type of starch can't be made any longer, and the peas actually, what happens, why are they wrinkled, is because when the plant can't make any starch in the normal way, it actually fills up with sucrose and becomes quite full of sucrose, and then lots of water comes in and it's got a high osmotic pressure. Then when the seed is drying down, when the seed ripens, all that water is lost and the seed becomes quite wrinkled. So that's why the seeds are wrinkled. There are other loci known or other genes known that will condition this difference between a round versus a wrinkled pea. Some of those loci genes are shown here. And any one of those will give a very similar phenotype for the same reason. And you can see some example of some of those on the left there. You can see some round seeds very obviously, and hopefully you'll be able to spot the wrinkled types in there and among those accessions. And these different types of starch mutants make different types of starch grains, which are very important in the food industry. They have different properties for making food additives. So another character that Mendel was studying that we know more about is the difference between short and tall plants. That's conditioned by the LE gene, which encodes a gibberellin GA3 oxidase gene. So yeah, plants, garden peas can be quite tall and easy for picking, but peas growing in the field are usually, well, they're always short because they can be easily mechanically harvested. So there's a really important difference there. And so, as I said, the LE gene encodes this enzyme that works at the very end of the pathway of gibberellin biosynthesis. So gibberellin is a plant hormone that regulates the plant growth. And so when you can't make the bioactive form of the hormone, which are the last 3 types shown here, then of course the plant is short. As you can see on the left-hand side of the panel there, these are actually isogenic lines differing only in the LE gene. And even at 4 weeks old, the difference is very apparent. Even younger than that, you can tell. And then it's even more apparent at 12 weeks old when the plants are quite tall. And you have to climb on a ladder to do the crosses. All right, another gene we know more about is the color of the seed. Now, green dried pea seeds, they can be yellow or green, and so the I gene conditions the difference between yellow and green seeds. And the reason for this is that the gene has a mutation in the magnesium dechelatase gene. And so what Why does this leave the peas green? Well, when peas mature, when they're fresh, they're always green in the pod. They're green in a green pod and the whole plant's green. But then the plant matures, it begins to senesce. And that natural process of senescence means that the chlorophyll in the plant is broken down. And the chlorophyll in the seed is also broken down in this way. So the chlorophyll pigment, when it begins to be broken down, the magnesium at the center of that ring structure in the chlorophyll molecules is removed. And that's the first step in the breakdown of chlorophyll. So when you have a mutation in that gene, you don't break down the chlorophyll stays there. So the seed remains green. And that's a really important trait. It's a quality trait for pea seeds. You know, you really want beautiful green seeds if you're making pea snacks, foods, things in cans. It's an important trait. Right. And so the last gene that we know quite a bit about is the gene that conditions the difference between white flowers and colored flowers, as I mentioned earlier, the A gene. And the pigmented flowers will also have a colored axil ring, and they will also have anthocyanin pigmentation on their seed coat sometimes. And so this gene is known to encode a basic loop helix transcription factor. And so in the biosynthesis of anthocyanin, we have a transcription factor complex that is known to operate, and it's conserved in many plant species. So on the far right here, you'll see this a representation of this transcription factor complex made up of a basic helix transcription factor, a WD40 transcription factor, and some mid transcription factors. As I said, the A gene encodes the basic loop helix component of that complex. And when it's mutated, the complex fails, and so we don't have transcription of the biosynthetic gene and enzymes that are required to make anthocyanin. And so we have a white flower. There's another gene also known that conditions this difference between white and pigmented. Not surprisingly, it's called A2, and it encodes the WD40 component of this complex. So when it's mutated, you'll also have a white flower. It'll look like an A mutant. You won't be able to tell the difference unless you do a cross. Right. So those are the ones we know about, and 3 we don't yet know what they are. So uncharacterized Mendelian genes, they look like this in the glass house. So let's start on the top left and look at a kind of a normal pea in the glass house on the far left. And, oh, sorry about that. And you'll see if you break open the pod, a fresh pod, you'll find inside that pod, there's a layer of lignin, and you can actually peel it back, as I've shown on the far left there. And that's great for shelling peas. You can easily break open the pod and shell the peas. That's good for field peas, but it's disgusting if you want to eat it as a mange too, because you'll be just like eating fingernails. Disgusting. Yeah. So there is a point in having P or V mutants, variants, which are shown in next door to that, and they don't have that lignin layer. So they're the ones that you'll get in the mange 2 peas. And you can see that because they don't have that lignin layer, you can see the outline of the peas inside the pod quite easily, as if they've been vacuum packed or something, because the pod is no longer stiff. Right, so if we move around now clockwise, you'll come to the fasciation phenotype here on the right. And these are where the flowers all develop at the same point at the terminal part of the inflorescence. They all come out at once, and the stem is very, very broad, compared to the normal way that peas grow, where the flowers produced at each node as you go up the stem in a very orderly manner. So fasciation type, the flowers are all there, and then all the pods appear at the same time too. So I think these type were quite popular in older times when you go to harvest your fasciated garden type and you just pull off all the pods at once and have a great big feast. I don't think many of us do that these days, unfortunately, but I think we should. So lastly, the GP trait that conditions a difference between green and yellow pea pods, as you can see here. And these traits, of course, have been genetically mapped a long time ago. So we know where the loci are distributed on the 7 linkage groups of P, as you can see here. And as I said in the previous slide, there are actually 2 loci that will condition a fasciation versus non-fasciation. And there are 2 loci that will condition a stiff pod versus a mange-tout type of pod. That's P and V. So as I said, well, actually there are some accessions of P that carry all 3 of these uncharacterized traits. So that's one of these is shown here. So this one, as you can see, has yellow pods. They have a kind of a vacuum-packed look about them. So they're either P or V. And then those pods are all appearing all at once. So it's fasciated. So it's going to be fast or far, but it's carrying all 3 traits. If we cross that with a normal P, make an F1 hybrid, a variable hybrid, and then self-fertilize that hybrid and obtain F2 progeny, sow them out in the field. This is what they look like. And this photo was taken just 2 weeks ago in the field research part of the John Innes Center. So these are segregating for the 3 Mendelian traits, and you can't see that in detail here. But what I want to actually show you in this picture is that this plot contains 1,000 F2 progeny. And I measured it the other day, it's 22 meters long by 5.5 meters wide. And funnily enough, that's pretty much the same size as Mendel's glass house, according to the plans. If you just take a look at the plans, there's a scale on the plans of Mendel's glass house, but you don't know what the units are. So I just did a rough calculation of what his glass house size was, and it is about this size. So that's the kind of scale that Mendel was working on. And that's just 1,000 plants. And he did, 8 years of this work and thousands of plants. So it's pretty impressive. And it's a lot of work, keeping them tied up, scoring the traits as they appear. They don't all appear at once. You've got to go there and check and go there and check and go there and check. You're a very busy person looking after these plants. So he would have had his avoir du poids in very good control while he was doing these kind of experiments. But as soon as he stopped, he might have got a bit bigger. Anyway, in that field plot, there's also growing a large number of plants that we are using for genome-wide association studies in collaboration with colleagues in the Aegis in Shenzhen. That would be the Agronomic Genomics Institute in Shenzhen, China. So yes, in 2019, P did enter the genomic era. It was a long time coming. It's an international collaboration that achieved this landmark, and we're very pleased that that happened. And I'll just skip over this editorial that was written in Nature Genetics with a little error in it. And well, since 2019, there have been improvements in genome assemblies, of course. And the first genome, which is called the Cameo version 1A, is shown in bleu in the middle. And then other assemblies, other genomes have come along since then. And you can see, according to the x-axis, which shows the number of contigs. That's the number of pieces the genome is split up into that you can't join together. The idea is to reduce the number of those. And also the idea is to cover as much of the genome as possible in your genome assembly, genome size being similar to a human, with a huge number of repetitive elements, about 80%, making it hard to join together. And so as you can see, the assemblies are getting better and better. So we're in a very good position now to know or have 7 pseudomolecules corresponding to the 7 linkage groups of P. So those are shown here. So now I'm coming on to, as you've guessed, at the unknown. And I just want to show you a picture, a snapshot of what part of a genome looks like when you're looking at it these days. So here I'm showing you 2 panels, a top panel coming from a green podded line and a bottom panel coming from a yellow podded line. And all of these tiny little marks that you see in front of you are short DNA reads, and they've been matched or mapped onto a genome, And so where they match, you can see they are in gray. That's a perfect match, and that tends to represent genes. And where they don't match, they're colored. That's where it's a bad match, and that represents retro elements, repetitive elements. And so that's what that represents. There's repetitive elements all over the genome, as I said, and they will match slightly to many different places. So you get this kind of colored distribution of retro elements all throughout the genome. But what's of interest to me here is this part here where we can actually see there's a very large deletion present in the yellow podded line. That's represented by just the complete blank you can see here in parts of the lower yellow podded panel. Right, so this is another version of that comparison. Top panel is the green podded line, bottom panel is the yellow podded line. And here we're just looking at genic sequences. We're not looking at the repeat elements at all. And so again, you can see that there's certainly a clear difference between the green and these 2 yellow podded lines below it. And the middle yellow podded line is actually an accession that was worked on in the 1920s at the John Innes Center by Caroline Pellew, who was studying the GP locus. And so we can see that this 100 kilobase deletion is present here. If we look in another line, which is the type line for this yellow podded phenotype, we see the same difference. And if we look in the entire collection at all of the yellow podded lines, and all of the known to be GP lines, they all carry this 100 kilobase deletion. And so that has been genetically defined, and the markers defining the GP locus are about 1 megabase apart. And in that megabase, well, is this 100 kilobase deletion, and not in that region are the candidate genes that were discussed by the authors in this paper here, Shirasawa et al. Last year, where they had candidate genes for the GP locus. Those are excluded from my genetic interval. So work here is ongoing, of course, actively. So that brings me to the other kinds of works that we're doing, and unlike humans, you can mutagenize Ps and generate lots of variants, and then investigate those variants if they carry traits of interest. And genomic sequencing, because you'll have a very isogenic lines in this situation, you can easily use genomic information to see where you've introduced a deletion by mutagenesis. And so you can see the deletion that's causing the the trait of interest. And so that's the kind of work we're engaged in at the moment. And so without further ado, I'll thank very much the members of both groups that I work in at the John Innes Centre, past and present members. So I work in the Domini Lab and the Ostergaard Lab, and all of the support colleagues at the John Innes Centre, the funding agencies, and of course our international colleagues as well. So thank you very much.